We're moving into the section on histology. So we'll introduce the primary four tissue groups of the body. And we're still at the microscopic scale. So in this case, histology means the study of tissues, cells, and their extracellular components. And that's what we're seeing here on this micrograph, this image here taken from the microscope. And this photograph is showing a segment of the small intestines. And in particular, it's a portion called the duodenum. And it really comes down to recognizing distinct shapes, such as these spikes, these villi, the staining, which is artificial, color has been applied. So it's truly just distinct coloring and shaping that I'm seeing here that allows me to recognize so many different unique features and determine that this must be the sample of the duodenum. And then there's various structural features that we'll later come to recognize. So on our spectrum, again, we're still at the microscopic level. And specifically, we're looking at the tissues here. And in our body, there are four principal groups of tissue. So we call epithelial tissues, connective tissues, muscle tissue, and neural tissue are also known as nervous tissue. But in any case, all of the tissues themselves are composed of unique types of cells and their extracellular material, and that combines to form the basis of these four general categories of tissues. So we're going to start with epithelial tissue, and just as we see here, in the brief description, covers the surfaces, lines internal cavities and chambers, and produces secretions. And that's where we want to start, is with epithelial tissue. And we'll follow this order here, moving into connective tissue, muscular tissue, and get, for now, just a very brief introduction to nervous tissues. So starting with epithelium, we find this tissue connected right at the surface of the body. And these epithelial cells are bonded together. So we have these sheets. You could think of it like a quilt. So a broad sheet of interconnected cells that cover our exposed surfaces. But it's not only our external surface, such as our skin and hair, but also any internal surfaces. So lining the inside of body cavities, the inner surface of any ducts and pore structures. So external surfaces, and lining the border of internal hollow cavities as well. That's where we find epithelium. So there's always that connection to the surface. And traits now that we find in epithelial tissues are spelled out here. And we'll continue with these on the next few slides. But epithelial tissues have this trait of cellularity. And this goes right to this trait here, that there's an interconnected sheet of cells. So we'll describe how epithelial cells are bonded to their neighbors. Also, since these tissues cover the surface, what holds them in place and connects them down to deeper structures is now what's known as a basement membrane. So this is what attaches the superficial epithelium onto the deeper surface of our body. Individual epithelial cells have this trait of polarity, lacking a blood supply. And with mitosis, we rapidly produce new cells to replace older ones. So we'll expand upon these traits. So this first one here, the trait of cellularity, we can make a little sketch. I'll just draw these two little boxes here. So these represent two neighboring epithelial cells. We're drawing the cell membrane, and their cell membranes of neighboring epithelial cells are now directly bonded together via various types of cell junctions. So this is how we get that sheet or quilt-like description for epithelial tissues. Their cell membranes are bonded together, and that's how we get this broad sheet of cells that's covering that exposed surface. It's now only on the very deepest edge of epithelial tissue is where we have that attachment structure known as a basement membrane. So if we add this to our sketch here, deep, of course, we know means away from the surface. The exposed surface is the superficial end, is one name for it, one term to describe that. And it's only towards the deepest end where we have that attachment structure known as the basement membrane. And this is what attaches the superficial epithelial tissue and bonds it down to the deeper tissues of the body. Another trait now of polarity, if we look at an individual epithelial cell, the opposite ends, and there's the term superficial versus deep, have distinct features. So we can still expand upon this sketch here. On the superficial side, we could draw in, for example, some microvilli or some cilia. Certain organelles may be found closer to the surface or deeper, and one example is already here. The basement membrane is only ever found towards the deeper end, and we're just describing that here. So the basal end, the deeper side of the cell, that's the only location where we find the basement membrane, whereas the apical or superficial, these are just synonymous terms. The apical end or the superficial end closer to the surface, again, there's various features that are only ever found towards that side of the cell. So with one cell that has these two very distinct ends to it, the basal side with its features, the apical end with its features, that's the description of polarity. The two ends have their unique structures. So here's some much better textbook illustrations to describe that. So the first one, to trait of cellularity, we can see how the group of cells here and their cell membranes are bound together via various types of cell junctions to make this broad sheet right here at the surface. We can see here on the deeper end of this tissue and expanding in the view here, we can see that attachment structure of the basement membrane. So that's what bonds these superficial epithelial cells down to the deeper layers of tissue. And this figure up here is much, much nicer. So again, the location of the basement membrane just to the deep end only. There's a uneven distribution of organelles. It's a good way to describe that. An uneven distribution of organelles, and especially only towards the superficial end would we find microvilli or cilia. So that's that trait of polarity. The trait of being now avascular means that there's a lack of blood vessels. So epithelial tissue does not have its own direct blood supply, but the living epithelial tissues do need oxygen, they need nourishment. So for that reason, epithelial tissue must be in close proximity to some other type of tissue which does have a blood supply. And that's usually connective tissue. And then finally, again, with the rapid pace of mitosis, we're able to now, compared to other tissues, rapidly produce new epithelial tissues to replace the older damaged epithelial cells of our body. So we have this pretty rapid turnover, and hair is composed of epithelial tissue. So we're able to grow hair as the older tissue breaks and detaches. It's being replaced by newer tissue. So we see these traits here in all of the unique types of epithelial tissues in our body. The cells are bonded together, typically attached to the basement membrane. There is an exception we'll see much later. The cells have this trait of polarity, so opposing ends of the cells are distinct, lacks a blood supply, and again, that ability to rapidly produce new cells. So now moving forward with epithelium, When we start to name specific types, we can see that there's now different ways that epithelial cells can be layered on top of the basement membrane. So we just pointed out the deep edge of epithelial tissue has that attachment structure of a basement membrane. So if superficial to that basement membrane, there's only one layer of cells, that is the description of simple epithelium. Just a single layer of epithelial cells. Each of those cells is attached onto that deep basement membrane. The term stratification truly means layered. There's multiple layers. So a minimum of 2, but of course there could be many, many more, 10, 15. But the point is, only the very deepest layer is attached to the basement membrane. And now all the other layers are stratified or layered, stacked, superficial to that. So showing that trait of cellularity, we have our basement membrane. The first layer of cells are attached to the basement membrane. And then with the trait of cellularity, cells are bonded together and they are stratified or they're stacked as they move closer to the surface. Another type of layering is called pseudostratification. And there's truly only one layer. But now due to the irregular shape of the cells, and I'll go to a photograph, some cells are longer, some cells are shorter, and that ultimately gives the appearance that they appear that the cells are stratified when they're truly just one single layer. And again, this figure here from your textbook shows. So simple layering, we want to find the basement membrane, and we just have one cell stacked superficial to that. The cells are attached laterally, not trait of cellularity, but there's no stacking. Simple layer, simple epithelium. Stratified epithelium, again, we can see the basement membrane. The deepest layer is attached to the basement membrane, and now all the subsequent layers are now simply stratified, layered on top of that towards the surface. And the photograph here showing the pseudostratification, and really what appears to be stratified, what's falsely stratified, is the appearance of the nuclei. So it does appear as if one nucleus is superficial to another, and that is the case for the nuclei. But when close inspection, every cell does contact down to the basement membrane. And here's that appearance of the pseudostratified layering. So typically when it comes to epithelial tissues, the layering, simple, stratified, pseudostratified, this is usually the first part of the name, describing how many layers there are. Another consideration now is the shape of the epithelial cell. So there's 3 basic shapes. There's what's known as a squamous shape. And the next 2 names are very descriptive, a little cube shape or a slender column shape. So cuboidal cells and columnar cells. And let's move forward with that. So the squamous shape, if we look from the deep end to the superficial edge, it's a very thin, flat cell. And when we view it, looking down onto it, there's no consistent pattern. So with that in mind, a thin cell, but a kind of a regular border shape, it always reminds me of just looking at a fried egg. You just drop an egg into a pan and just takes on some shape, but it's quite thin. That is a squamous shaped epithelial cell. A cuboidal shape, again, just like the name implies. A little boxy cube is the appearance under the microscope. So that's how it views through cross-section and gets that appearance. And we can see those here brought around into a ring. And then lastly, the slender, tall, columnar shaped cells. So there's much more length as compared to their width. So these slender columnar cells. And again, the shape of the cell is usually the second part of the name. The third part of the name is now just the tissue group. So we'll put this together. When we want to name specific types of epithelial tissues, we typically first start with the layering mechanism, simple, stratified, pseudostratified. The second part of the name is the shape, squamous, cuboidal, columnar. And then the third part of the name is the category. So we could say epithelial tissue. So here's just some examples. Based on just the name that you see here, this is really a descriptive of what we see. So one layer on the basement membrane of the flat, thin cells, that would be called simple squamous epithelial tissue. Here we're seeing one layer superficial to the basement membrane of the cube-shaped cells, simple cuboidal epithelial tissue. If we have multiple layers, so if we had 2, if we had 5, stratified, cuboidal, and here, if you use the term epithelia or epithelial tissue, epithelium, there'd be no difference there. All right, so that's how we name specific types of tissues, typically with the layering, the shape, and ending with the tissue group. But there are some unusual cases that are outliers that we'll see once we get to those specific areas that have them. So now we want to go back to look at these traits, the layering and the shape. There's benefits for each. So we'll go back here to simple layering, a single layer of cells. The benefit here, this is great for diffusion. It's just a very thin barrier, one layer of cells only, so diffusion and absorption through that single simple layering. So for example, the tissue in your lungs are made of simple epithelium, and gases could diffuse through. Stratified layering, having multiple layers, the benefit here is protection. So having multiple layers of cells stacked up, you could damage the superficial layers, but now there's all the deeper layers providing all of that additional protection. So our skin is a great example of the stratified layering. Pseudostratification, and you notice we'll use the figure here. We typically see pseudostratification along in concert with these epithelial cells having cilia. So with this arrangement, the pseudostratification with the cilia, which is the typical case, the cilia move and any material that's at the surface of the epithelial tissue or material that's at the surface, as the cilia move, we could propel material across the surface with the sweeping action of the cilia. So propelling material across the surface. And an example would be your trachea, your windpipe. The internal surface of our trachea has cilia, and if we inhale any material, something gets trapped in that area, and typically just mucus as well, it'll sweep using the cilia to propel that material out of the airway. There's the shapes. The benefit of the different shapes, the flat, thin cell, this is great for diffusion. So again, just a very thin barrier for material to pass through. The cuboidal cells that we're seeing here, cuboidal cells form multiple ducts and pores of our body. So some duct structure or some pore. And that's made of a ring, but the ring itself is made of individual cuboidal cells. So now that we have this duct structure, we can now transport fluid through that duct. So if you notice here on this textbook figure, of course, they're illustrating here the pores of a sweat gland as an example. That's what I was trying to draw. The fact that the pores themselves are made of a ring of cuboidal cells, and the duct is used to transport secretions, transport fluid. And then finally, the columnar cells, such as our intestines, a great example. These are great for absorption. So for example, the nutrients that we eat and we could absorb in the area of the stomach and intestines, and absorption of material, columnar cells have that benefit. So when we get to lab portion, we will see examples of simple layering, stratified layering, pseudostratified layering, and we'll see the three different shapes. All right, moving forward with epithelial tissue, we're now going to see the functions. So what's the activities of epithelial tissues? Up to this point, we've talked about locations and shapes. And now we're going to see now this exocrine function. So exocrine secretion, this means that some type of fluid material is being produced, and that fluid secretion is released to the surface of the epithelial tissue. So it'll be released so that it'll coat the surface, whether it's external surface, such as your sweat glands, your oil glands, or an internal surface, such as your salivary glands, releasing saliva into your mouth. And any type of glandular structure of the body is derived from epithelial cells. It's the epithelial cells themselves that are producing the secretion for that gland, and then the secretions have to be released to the surface. So in terms of the fluids produced, there's these three primary types of glands. So this first one, we've used the term before, a serous membrane. Serous membranes, such as visceral and parietal membranes, contain serous glands, the source of their fluid, mucus, and then the combination of the two is truly what's known as a mixed exocrine gland. So we'll continue there. A serous gland is producing a serous, watery secretion, so it's very fluid. A mucus secretion, just think of the mucus in your nasal cavity and your sinus cavities, very important to your trachea and digestive organs. And mucus secretion is much more viscous, so a much thicker viscous secretion. And a mixed exocrine gland is truly one gland that produces the combination of both serous secretions from some cells, mucus secretions from other cells, and of course, it combines, it mixes, and that's what's known as a mixed exocrine gland. Our salivary glands, the production of saliva, is a great example of a mixed exocrine gland. So of course, we have these different types of fluid secretions, and all of which are now being produced by the epithelial cells of the gland. The secretion is produced internally within the epithelial cell. So you can imagine if we're looking at this gland here, this epithelial cell is producing some type of fluid secretion. Is it saliva or breast milk, whatnot? We have to now release that secretion out of this individual cell and allow it to pass into the duct. So we have three different mechanisms, three different methods of releasing that secretion out of the cell, past the cell membrane, and into the duct. And that's what these three processes here are. They all have the same result, releasing secretions from the cells, but it's their mechanism, the process differs between these three. So we'll start with merocrine. This is the most common of the three mechanisms. And we have a description of the process. So in this Merocrine process of secretion, the secretions are first produced and packaged in what's called the secretory vesicles. And numerous secretory vesicles are being produced here within the cell internally. They're packaged with now droplets of that secretion. And as the arrows show, these vesicles now migrate and contact the cell membrane. And once they fuse onto the inner surface of cell membrane, that opens up a small pore, and the contents of that vesicle can now be released into the duct structure. There's a nice textbook illustration covering all three modes of secretion. And this is just a larger image of the same thing. So the Merocrine process, using multiple secretory vesicles, it's just a much bigger figure here. And as that vesicle merges with the cell membrane, opening up that small window, and the contents can now flow into the pore. A second way to allow for this release of product is with the apocrine secretion. In this mode of secretion, first there's a buildup of vesicles just near the apical end of the cell, closer to the surface. And once there's a buildup of multiple vesicles, then a large portion of the cell simply detaches. It's shed off. And now all of the contents that were built up in this region is now flowing into the pores. And of course, there's little fragments of the cell itself. So this does damage a portion of the cell, but the cell can repair itself and do this process throughout its lifetime. So an example of a gland that uses this process is now mammary glands and their release of breast milk. Still a third mechanism, and again, there's a great figure in the textbook, the holocrine mechanism. The cells are now building up so much fluid secretion that in the first step, these cells are building up so much secretion that they start to swell up, and they continue to enlarge and swell up with more and more secretion to the points where the cells actually burst. So when the cell ruptures, killing the entire cell ruptures, and all of the contents of the cell are now flowing into the pores. And this is done in what's known as a sebaceous gland, uses this rupturing holocrine mechanism. And for now, we'll just mention the sebaceous gland. This is a oil-producing gland of our skin. And the cells of that sebaceous gland are rupturing, releasing oil ultimately, as you see here, right into the hair follicle, and in turn, it leads right into the surface. So we have developed three different mechanisms, but again, they all have the same goal of releasing the contents from the cells to get the contents into the pores. And that's where we'll finish with this first tissue category of epithelial tissues. We're moving into now the category of connective tissues. And like the name implies, this is a connective or attachment support type of tissue. The other three categories of tissue are very well defined and unique types of cells. And truly connective tissue, if the cells don't fit into the other three types of categories, then they're just simply lumped into this category called connective tissue. And if we take a look at the diagram here, you see that there's a very broad diversity among connective tissues. So there are these three subdivisions, and we'll see the details of each of these. which are then further subdivided. But in this very broad category, we have, for example, adipose tissue, we have collagen, blood, cartilage, bone, all lumped together in this one category of tissue, totally known as connective tissues. But what they do have all in common are a few defining traits, and we'll start with those. So within connective tissue, there's various types of fibers. You see these three primary groups of fibers, these strands of protein. All of these different types of protein fibers are produced by a cell known as a fibroblast. And we'll describe the cells shortly, but we want to focus first on the fibers. So these three primary types of fibers, the collagen fibers, reticular fibers, and elastic fibers, are found in various types of connective tissues. So we'll start with collagen. The collagen fiber itself, an individual strand of collagen, is actually like a braided protein. So you can think of it like a rope. And just like a rope, it's quite strong. So any connective tissue that has collagen running through it, and the more collagen you have, that'll make that tissue stronger. And a great example of where we find this, you notice the textbook figure here, but tendons and ligaments are great examples of structures that are composed of just a large amount of collagen fibers. So these strong tissues here. Second type of fiber, reticular fibers. In this figure, they're seen here as these thin, branching, black fibers that are stained here. And that's their appearance. So these thin, branching fibers. And they start out as just the basic structure and framework for many, many organs, beginning with this internal lattice work of reticular fibers. And then in turn, the actual cells of that organ are now attached to that lattice work. So an example would be a liver or a spleen. Those are just some examples of organs that are rich in that lattice work of reticular fibers. And if we make a little sketch here, just the basic shape and structure of the organ begins with the branching frame of reticular fibers. And then in turn, the actual cells of that organ are now attached and suspended within that three-dimensional framework of reticular fibers. The third category, third type, elastic fibers. These now allow for areas to be stretched and distorted. And after it's stretched, it then can recoil back to shape. So the textbook figure is showing what's known as elastic ligaments. And these are a good example. But I think a more obvious example would be a blood vessel. So you can imagine an artery, and every time blood is pumped into the artery, that artery will expand from the pressure. And with the high level of elasticity, it then rebounds back to shape. And so it's with these three types here, elastic fibers allowing for the tissues to be distorted and stretched. Reticular fibers, that branching lattice work, making the shape of the organ, and then simply just the strength of collagen. But all three types of connective tissue fibers are made by these cells, and the cells that produce fibers are fibroblasts. So now we'll move on to cell types found within various types of connective tissues. So we're going to start here with connective tissue proper. So we're limited to just this sector here for now. And we'll see a few types of cells that are present in connective tissue proper. So it's now divided into what's known as fixed cells is one category. And a second category we'll get to on the next slide coming up. So what's meant by fixed cells, these are the common typical cells that we find in connective tissues. So the usual players that are found, and the first one makes perfect sense. Anywhere that we're going to have fibers, collagen, elastic reticular fibers, we need the fibroblast cells that produce them. So it produces and secretes that material which becomes the fiber itself. There's what's known as a fixed macrophage, so a white blood cell that's embedded within the connective tissue. White blood cells, of course, have that immune function to fight off foreign infection, foreign material. An adipocyte, commonly called a fat cell, doing its job of storing fat, storing it. Melanocyte cells, these are cells that produce the pigment melanin, so adding color to the tissue. So you can think of melanocytes, of course, giving pigmentation to the skin, color to the iris, so pigmentation of tissue. And this last one, a very important type, a mesenchymal cell. This is a connective tissue specific stem cell. So mesenchyme, this is the way that we produce and replace connective tissue cells. Mesenchyme cells can now develop and differentiate to make new adipocytes, for example, to make new fibroblast cells. It's our way of producing new and replacing old connective tissue cells. So for example, here with the sperm and the egg, we have what's called an embryonic stem cell. And from an embryo, that fertilized egg has to make every type of cell for that baby that's developing. Whereas now a mesenchyme cell is more limited. Mesenchyme cells can only produce distinct connective tissue cells. So now the second category beyond fixed, there's now wandering cells. And what these all have in common, wandering, these are cells that actually travel through the body, traveling through our cardiovascular system. The wandering cells enter into the tissue only when there's a need, if there's an infection, if there's an injury. So when we look at the specific examples here, free macrophages, lymphocytes, macrophages, and you see that similar structure, that similar function, responding to pathogens, something foreign, having this immune function, and even down here, once again, immune function, because these are truly just different types of white blood cells. And the same would go for a mast cell as well. Mast cell, a very important part of our immunity is, first of all, inflammation, increased blood flow to an area, increased delivery of white blood cells, healing factors. So again, these cells travel through circulation. We're truly just describing various types of specific white blood cells. And here we're seeing just a brief description for the different examples. There are 5 different categories of white blood cells. And here we're seeing now some of the various examples. So when there's an injury, when there's an infection, when there's a need, these travel through the bloodstream, and these cells now leave the bloodstream and enter into that injured or infected connective tissue. So now we're going to move into the categories of connective tissue proper. There's both the loose and the dense. And we're starting here with the loosely arranged connective tissue proper. So this is a great cushioning, packing, protective type of tissue. And what we want to focus on is locations, where we find these. So 3 examples of loose connective tissue proper. There's areolar connective tissue, adipose connective tissue, and reticular connective tissue. And we're just describing locations where we find these. So some examples, areolar is especially common type of connective tissue. So we find this in the more superficial portion of our dermis. It's a cushion around many different organs. So blood vessels and nerves, just a general cushioning protective tissue. The same goes for adipose tissue. We have this deep to our skin in what's called the hypodermis, or the subcutaneous fat. There's a cushion of adipose between your eyes and the bones of the skull. And adipose is principally the cushion that supports and protects our kidneys. Whereas now the reticular tissue, and you want to think back to those branching reticular fibers, making the basic shape of organs such as the liver, kidney, the spleen, even the heart. And again, locations, that's what's seen in the textbook figures. So I want to point these out to you. And you got a nice illustration as well of what these loosely arranged connective tissues look like. And truly, the description of loose means there's a lot of spacing between the fibers. So a loose arrangement of the collagen fibers or elastic fibers or reticular fibers. And in life, the spacing is filled with fluid known as ground substance. There's now the dense connective tissue proper. So this implies that the fibers, the collagen fibers or elastic fibers, are now densely, closely packed together. So we're looking at 2 of the 3 examples. What's known as dense irregular connective tissue versus dense regular connective tissue. Both of these have a dense packing of collagen fibers. So the strength of collagen is present in each of these. Dense irregular connective tissue, the collagen fibers are now woven together in just a random pattern. We find this in the deeper portion of the dermis, for example. So the deeper area of the dermis. Whereas dense regular has a dense packing of collagen fibers. However, the collagen fibers just run roughly parallel to one another. So that's the difference there between the irregular arrangement versus regular arrangement. And we see the locations for dense regular, tendons and ligaments are the great examples. This term aponeurosis is truly just a distinct shape and type of tendon. So now we'll move forward to the next category of connective tissues. So what's known as fluid connective tissue. These are now connective tissues with a large amount of water, a large amount of fluid, and suspended in that fluid, of course, are the various cells. So for now, we're just going to bring up the 2 examples. So the 2 types of fluid connective tissue, there's blood, the fluid of our cardiovascular system, circulating through the heart, the blood vessels, the capillaries, erectile tissues. And that's the figure you see here. And a second type of fluid connective tissue, lymph, unique to the lymphatic system, our lymph nodes, our lymphatic vessels. And we'll cover each of these fluids in more detail at a later date. when we cover these organ systems. There's now a third category of connective tissue known as supporting connective tissue. And supporting connective tissues include both cartilage and bone. So these two types are very distinct from one another, cartilage and bone, but they are grouped together in the same subdivision known as supporting connective tissue. So we'll start with cartilage. All forms of cartilage are this flexible yet firm avascular gel. So lacking a blood supply is the typical and usual condition for cartilage. We'll see an exception coming up. And of these three types, hyaline cartilage, this is the most common type of cartilage. And again, discussing locations for connective tissues. Hyaline cartilage, this is found between the ribs and the sternum. The breastbone is that sternum. The windpipe, the trachea, you could feel that hyaline cartilage right on your windpipe, on the trachea. And it's the type of cartilage that's in our joints. And you can see that, of course, if you have a chicken leg, for example, you can see that clear gel at the end of a drumstick. Fibrocartilage is a much stronger type of cartilage due to the increased amount of collagen running through it. And one example of a location, the symphysis pubis, that's the fibrocartilage of the pelvis, as well as the discs between our vertebrae. The intervertebral discs are composed of this stronger fibrocartilage. And the last type, elastic cartilage, this elasticity is afforded from the elastic fibers that run through this type of cartilage. So making it extremely flexible and allowing it to recoil once stretched and distorted. So here's that term otic, right? Otic, of course, meaning the ear, as well as the cartilage of our nose. So this avascular gel. However, now in the same category, bone has an extremely rich blood supply. So bone is highly vascular. And again, we'll get more about bone tissue in a later chapter. But the major contributing factors for bone tissue is, again, the same collagen fibers we were just describing, and the deposits of calcium. So making the bones much, much more rigid. And again, the textbook showing examples of locations. Hyaline cartilage seen here in our joint is a great example. Elastic cartilage in the ear. And fibrocartilage, those intervertebral discs, just for example. So now we're moving into a third category of tissue, muscular tissue of the body. So all of our muscular tissues have the ability to contract. So these cells can reduce their length as they shorten and contract. And this is used to produce and generate a force. So the three principal types of muscular tissue, there is skeletal muscle, cardiac muscle, and smooth muscle tissue of the body. So beginning with the location for each of these, skeletal muscle tissue, as the name implies, it's attached on to our skeleton. It connects to our bones. So the various tendons of our muscles, those tendons connect onto the bones. That's the location for skeletal muscle. Skeletal muscle now has these two different systems of control by the nervous system. So our nervous system, our central nervous system, the control center for all of the functions of our body. And there's both what's considered voluntary control and involuntary control of skeletal muscle that comes from the nervous system. So a description of this voluntary control, you could say conscious at will control. If you just want to wave your hand, you just simply think of doing that motion, and you can actively and consciously control that contraction of skeletal muscle to wave your hand. Whereas involuntary control, this is now where skeletal muscle will be stimulated, but it's not at the conscious level. And a great example of this would be breathing using your diaphragm. And again, skeletal muscle has both voluntary and involuntary control. And your diaphragm is a perfect example. If you decide to take a deep breath, you could breathe in, take a deep breath using your diaphragm. But of course, you could be completely unconscious, asleep, and your diaphragm will still contract to keep you breathing as you're in sleep. So that involuntary control. Skeletal muscle has both types. Cardiac muscle, this is exclusive to the heart, no other organ, just the heart. And cardiac muscle only has involuntary control. So again, our nervous system could speed up or slow down the heart rate, but it's at the subconscious level. So if we don't have that direct conscious level of control, we can't simply sit in a chair and decide that you want your heart to beat faster. However, if you are more active, if you're running around using your skeletal muscle, as a result, we have that involuntary effect to speed up the heart rate. Smooth muscle, this is found in many hollow organs. That's kind of the general description. Hollow organs such as your stomach, your urinary bladder, arteries and veins. So a lot of hollow tubes. In the wall of that tube, there's a layer of smooth muscle. And again, this has involuntary control. And to stick with that example, we could use the iris would be a great example. You could concentrate as much as you want, but you can't dilate your pupil. However, if there's a change in light levels, that could lead to that involuntary or that subconscious control to now change and adjust the size of the pupil. So we want to focus now on this chart here. And you notice I have these different sections boxed in. We want to add a few more points of information about muscle tissue. So we know the three types: skeletal muscle. This is, of course, located throughout the body, connected to the skeleton. Cardiac muscle, exclusive to the heart, nowhere else. Smooth muscle in multiple hollow tubes, such as blood vessels, the digestive tract. Second trait we want to point out here is now only cardiac muscle has these unique types of cell junctions known as intercalated discs. The other two types of tissues do not have these present. So intercalated disc unique to cardiac muscle. And we can see it here on this figure. So this cellular connection between neighboring cardiocyte cells. And now using these figures as an example, we could discuss the appearance of the cells. So in skeletal muscle, we notice that we see the striping, known as striations. They're once again present in cardiac muscle, more of those striations. However, smooth muscle lacks the striations. So we'll say the stripes, and the stripes are present in skeletal muscle and cardiac muscle, but they're absent from smooth muscle. And another trait we'll take a look at is the number of nuclei. So how many nuclei are found in one cell? So in skeletal muscle, and we're only seeing one little small segment, but in one skeletal muscle cell, there's actually multiple nuclei. So we'll just say many. There are dozens of nuclei in one cell. Cardiac muscle cell, it usually only has one nucleus per cell. Sometimes, the rare case, they'll have two. And in smooth muscle, there's only one nucleus per cell. Final trait for now about muscle tissue. In order to contract, all three types of muscle tissues rely on an energy source known as ATP. And that's the energy source that we're using to contract and for many other functions in the body. Well, it's a source of energy. So all muscle tissue has that in common. And the last type of tissue that we're covering, the fourth tissue category, it can be called neural or nervous tissue. And the unique traits now for this tissue, nervous tissue has the ability to rapidly send and transmit these electrical currents throughout the length of the cell. From one end of the neuron to the opposite end of the neuron, we do transmit this electrical impulse. The huge majority of this tissue is found just in two organs. The brain, the adult brain, develops to about 3 pounds of nervous tissue. The spinal cord has about 100 million cells. So that huge majority just found in these two organs. And within nervous tissue, there's two categories of cells. The neurons, these are the ones that are actually sending these electrical impulses. So they do the work of the nervous system, transferring information, processing information once it's within the brain or spinal cord. And the neuroglial cells, in some way, they aid and support the function of the neurons. That's their job, is supporting the function and health of the neurons in some fashion. And then finally, you see the ratio. The neuroglial cells are more common. So truly, a neuron is covered and surrounded by various types of neuroglial cells. And again, we'll get more about this tissue category once we study the nervous system.